It's my pleasure to welcome Mark Strauss to the EBI today. Mark is a professor of microbiology and the Campus Alberta Innovates Chair at the Department of Geoscience at the University of Calgary. He obtained his PhD at Delft University of Technology in the Netherlands, working with Yves Quenin. And during his project, he pioneered the microbiology of anaerobic ammonium oxidation. So it was one of the missing lithotropies, which were postulated back in the 70s. And Mark has probably more than anyone else contributed to identifying these missing lithotropies over the years. So he's also done work to identify the microorganisms which can couple methane oxidation to nitrification. And both of these discoveries originated from just fundamental thermodynamic inferences that these metabolisms must exist. And he's now transitioning some of that work to thinking about how thermodynamics control the structure of microbial communities and how communities are selected for the environment. And he has been at the Max Planck Institute for Marine Microbiology in Bremen, but has moved a year ago to be in Calgary. And his many scientific discoveries have received national and international recognition in the form of papers in high-impact journals, a membership in the Young Academy of Sciences in the Netherlands, prestigious European research grant, and the ISME Young Investigators Award, among other honors. So without further ado, Mark, thank you. Welcome to EBI.
All right, thank you very much for a nice introduction. I'm happy to be here. I didn't put it on the slide, but when I started in Calgary, I named my group the Energy Bioengineering Group. And actually, I didn't realize that your institute was named very similarly. So I guess there is some synergy or overlap or shared interest between what we are doing. So that's also why I'm happy to be here now. So this presentation is entitled "The Power of Microbial Communities," and I'll just indicate why it's called that way. So the example, use of microalgae for bioenergy production, that's one of the projects I started when I was working in Germany and following up more intensely now in Calgary. So the common approach worldwide seems to be to bioprospect or genetically modify strains of algae to have favorable properties, for example, high lipid content, to be used for biofuel production. In my opinion, production of fuel is really the kind of basis of the economy. It's really a bulk product, and it needs to be produced at really the lowest costs. And when you think about using essentially pure cultures for this purpose, I think they lead to at least a necessary cost. And those costs are related to you're taking the culture, you're scaling up, you're running the process of almost unimaginable large scale when you think about really making a significant contribution. So you get a lot of evolution in there. You may get contamination. This morning I learned about the risks of phage infections. If you genetically modify, things may not stay the way you intended it. And I put the image here of Yoda, may the force be with you. Essentially the problem is that by doing this, you place an organism in a ecological context where it would never be in a natural ecosystem. It's not with the selective force. It's against the selective force. And when you go against the selective force, you run into problems that lead to higher costs. So that's what I mean by the power of microbial communities. If you would use a natural community for this purpose, you would not have those problems. And I think there's a very nice paper on this concept by one of my mentors during my PhD, Mark van Loosdrecht, and his colleague Robert Kleerewezen in Delft, about how the theory behind this. And for example, so essentially they say, well, you shouldn't engineer the organisms, you should engineer the environment in such a way that the organisms do what you want to. And for example, Robert Kleerewezen is a big specialist in fermentation, and thermodynamic analysis of fermentative systems, and he tells me that if I define a system for him, the conditions, the environmental conditions, the substrate, he can predict me exactly which fermentation products will result. So fermentation is close to thermodynamic equilibrium, so relatively predictable. They also developed a process for microalgae cultivation based on this concept. So what they said, okay, we want a process that has a lot of lipids. So what they did was say, okay, what we do is we only provide the nutrients, so ammonium, at night. So what happens is during the daytime, the organisms have no way of producing protein, so they store all their energy in lipids, and then during the nighttime, they convert those lipids into protein. So when you harvest them just before it gets dark, you get a very highly enriched, lipid-rich material, even though you didn't use a strain that did that from the start. Now, in this presentation, I will not talk much about my ventures into the algae business because we're still working on the IP, but I'll give you my history and how I came to these communities. And when you want to work with communities, you have to understand kind of the rules of the game, the rules that shape how these communities and what kind of processes you enrich. Because if you don't have those rules, you don't know how to kind of entice or force the community to do what you want. Now, one of those rules is all the pathways that we know, right? So you cannot expect microbial communities to do things that cannot be done. And here you see an example of the biological nitrogen cycle as it was known in the, I think probably still in textbooks in the 1990s, early 2000s. The other, I think probably most powerful predictive rule in microbial ecology is the microbial redox tower. Yeah, so essentially this tower, as drawn here by Fenchel and Jurgensen, this tower says the different electron acceptors, so oxygen, nitrate, manganese, iron, sulfate, CO2, are consumed in their thermodynamic order. Now that's really very central to our field. So there is some role of thermodynamics in it. So these are examples of rules that you need if you want to address microbial communities. Well, and I'll show you, and I probably will not get to the final part, but both of these rules are wrong. Yeah, so were shown to be wrong. So the challenge at the moment is to do the fundamental research and redraw all these things as they really are, because otherwise your communities are always going to give unsatisfying results, and you may be tempted to go to resort to genetic engineering. Now, most of my past research was on the biological nitrogen cycle, and I think this cycle is very important also to much of the work that is going on here. And what you see on this graph is that we make a very significant contribution to emissions of carbon dioxide to the atmosphere. It's more than 5% of the natural background. That's very significant. Difficult to show, that's all the debate is about, but significant. When you look at the nitrogen cycle, the situation is really very much worse. So when you look at the contribution of fertilizer alone, we are already almost at the natural background of fixation of nitrogen from the atmosphere. And when you add the deposition of ammonia from fossil fuel burning, we're actually on probably about equal. That's a very significant human influence on the nitrogen cycle, leading to a lot of ammonia and nitrate in natural ecosystems. And when you consider that most of this nitrate is removed by denitrification, then and denitrification produces nitrous oxide as a byproduct. Well, nitrification also produces nitrous oxide, and anaerobic ammonium oxidation also. And according to the global budgets, this is about 1% of the fertilizer we put in the field, because the warming potential of nitrous oxide is so strong and its atmospheric lifetime is so long, this may offset any benefits you gain from biofuels if you need to fertilize. So really important cycle. So then we go to so just give you a little overview of my past work with communities. So this is the city of Delft, where I did my PhD work as a chemical engineer, and in the center of this very old medieval town, there's a yeast factory. It's about here, and the yeast factory in this town of 80,000 inhabitants produces the wastewater of a city of 1 million. And so it's right in the center. There's no room to do any wastewater treatment. They already had a big odor problem because of the yeast production. So they had to do something funny to treat their wastewater. I mean, it was untreated until the beginning of the 1980s. So that's pretty amazing because this all went into quite tiny river. And what they did was they tried to remove sulfide by denitrification. And the advantage to them of the denitrification was that they didn't have to aerate their system. And so they didn't have to be afraid that somehow a lot of hydrogen sulfide got into the air and contributed to their already bad reputation. And then Arnold Mulder, the engineer who was running the pilot plant, noticed that in that bioreactor that was oxidizing sulfate with nitrate, what he saw over time, the ammonia also went missing. And he knew his literature, so there was this prediction by Engelbert Broda from the 1970s, says, well, actually, even though according to our beliefs, ammonia needs to be nitrified, we need oxygen for that. Actually, it should be very well possible to do that with nitrites or nitrates. And that started this whole research line of anaerobic ammonium oxidation, where I started as a PhD student in 1995, and my task was to apply this process in industry. So it was a really applied project to make it work. And it was known at the time that by work of my predecessor, Astrid van der Graaf, that probably this reaction went as follows. You had nitrite and ammonia, which was combined into hydrazine and then oxidized to the nitrogen. But it was a very troublesome project. She spent probably, maybe half of the time of her PhD trying to isolate this organism, which never worked. And my contribution as an engineer was that I further develop these cultivation systems to grow these organisms. One of the most important problems was that they divided only once every 3 weeks. So that's one of the explanations why it was not so easy to get into pure culture. But this was a mixed culture. So it was a microbial community in a bioreactor fed only with ammonium and nitrite, and the selective force led to the enrichment of these organisms. So what I did at the time, and the microbiologists found this very difficult to accept. It was before, the whole metagenomics, fish was just starting. It was somewhere in limbo, this research, but what I did was I purified these dominant organisms from these bioreactors in a density gradient and showed that they were by themselves capable of doing that. And that kind of made everybody say, okay, well, it's maybe real after all. And so these organisms were later shown to be related to a group of microorganisms known as the planktomycetes, or now also called planktomyces Furucomicrobia chlamydia supervi. Okay. Now the research continued, and with the advent of genomics and proteomics, it became possible to really go into the molecular details of this organism, even though it was not in pure culture. And it turned out that its molecular machinery is really highly, highly complex. Inside this organelle in these cells that is called the anamoxosome, and it has many, many different cytochrome C proteins that are apparently all required to perform this metabolism. It's almost like it reminds me a little bit of a nuclear fusion reaction. Apparently the organism needs a lot of enzymes to keep this reaction under control. And one of the recent papers by Malk et al. Showed the crystal structure and the functional role of one of these enzymes, which actually appears to have nothing to do with this pathway. What this enzyme does is it oxidizes hydroxylamine to NO. And so probably one of the reactions necessary to make this going. So really complex thing. So I'm not a biochemist by training, and so for me, after, let's say, applying this in industry, so you can save a lot of money by applying Animox. This is the first Animox plant constructed in Rotterdam, the Netherlands, at the wastewater treatment plant. And you see it took many, many days to get going. And so really, again, because of this long doubling time. But in the meantime, many of these wastewater treatment plants have been built. And when you calculate, it's actually quite an amount of CO2 that is being saved, or energy that is being saved on a global scale by this process. So it's very nice, and it shows that the textbooks are maybe not the best place to look for rules for communities. And of course, everybody said, well, if it's so slow, it cannot be important. But thanks to the group or the work of Marcel Kuipers and colleagues, and followed up by many others in the meantime, it was shown that actually, even though unknown and not noticed for decades, actually it contributes maybe even 50% of the nitrogen in the atmosphere. So even though something is not known, it does not mean it is not significant. Okay, so as I said, I'm not a biochemist, so what I was interested in is whether this approach of Engelbert Broda and saying, okay, if an organism should exist thermodynamically, does that mean that it really exists in nature? So one of these other organisms, so I drew a little periodic system, difficult to find the spot, of electron acceptors and electron donors, and match them. What do we know? Those are the green spots. And what do we don't know? And right in the middle is this combination of using nitrate to oxidize methane. So people that looked for it, never found it, had been dismissed. It's probably impossible because the methane needs methane monooxygenase to activate. So it's not surprising that you don't find it. Well, with sulfate you find it, but that's another biochemistry altogether. There's always an explanation for something you don't find, but who knows? And I thought, well, maybe it wasn't found because it grew slowly. So at the time, with my PhD student and collaborator, Ashna and Katinka, went to a canal where you had quite some nitrate, agricultural runoff, and quite some methane, sampled the sediments, put it in a bioreactor. Same as for Animox, again, allowing the cells to grow very, very slowly. And Ashna, during her PhD, managed to really find this process. And now I'm going to skip over it a little bit fast, so we do fast forward now. So in the beginning, it looked like it was a consortium of archaea and bacteria, like for the anaerobic methane oxidation coupled to sulfate reduction. But in the end, it turned out the bacterium could do it by itself. And again, this was a phylum without any cultivated representatives called NC10. And this dominant organism in our culture was part of that phylum. And of course, there's many other organisms from different places that are also in this phylum. I think the organism was identified, or this culture was gotten in 2005, and in 2010 the whole mechanism had been solved. So metagenome had been done, and we were very lucky in this case that the genome of the dominant organism could be closed. So that made things very simple. And first the results looked a little bit frightening because this organism did not appear to be capable of denitrification, as it misses the final step of the denitrification pathway. And also what was frightening was that it had the complete aerobic methane oxidation pathway. So at first we thought we made a big mistake with these cultures. It was probably oxygen coming in, but and it kind of led to a lot of confusion for a while until somebody suggested that maybe the NO was dismutated to oxygen and nitrogen, which is a reaction that is thermodynamically feasible. It was actually part of a British intelligence program because they were afraid that the Russians would poison the atmosphere by doing the reverse reaction in the middle of the Cold War. But this direction is the thermodynamically favorable one. And Katerina Edvig, who followed up from Ashna, was the one who, after many experiments, managed to show that indeed oxygen was produced by this organism. So she really had to give it a very hard time by shutting down the methane monooxygenase with acetylene and then using 18O nitrite to produce 18O oxygen. So that showed the oxygen production. It was not possible experimentally to do this directly with NO. And maybe the fact that oxygen and NO react quite vigorously chemically was a problem there. Now later, when I was at the Max Planck Institute, there was a PhD student of Frits Widdel, Johannes Sedelius, and he enriched a gamma proteobacterium that oxidized alkanes, and it used nitrate as an electron acceptor. And it didn't use the fumarate addition system because its genome was known. So they were looking for a mechanism. Of course, they thought, well, maybe it's using the same mechanism. So what I did was I compared the two genomes and looked for a gene that they might have in common. And doing a little bit of comparative genomics, and right on the top of the list was this kind of strange form of NO reductase. So NO reductase, nitric oxide reductase, is part of the same superfamily as oxygen reductase. And so it's the same type of configuration of metals in the membranes in our own mitochondria, the heme, copper, oxygen reductases. And but these two enzymes had a really drastic modification to their active site. And in both organisms, they were among the most highly expressed enzymes. I think based on those data that this enzyme is the most likely candidate of catalyzing this NO dismutation. So that is the missing biochemistry here. And now we come to a kind of redefinition of the nitrogen network. And so this is kind of the rules rewritten. We now know that the situation was a little bit more complex than we thought before. And when I started in Bremen, what I wanted was to understand, how do I know when I supply an ecosystem with nitrate, what happens to it? Which pathway will now become important depending on the condition? And you see that 3 of the 4 pathways in this graph all lead to nitrogen. So that is the same ecological consequence, right? The loss of the nitrogen from the ecosystem. There's one pathway, which is already known for a long time, that goes to ammonia. And that, let's say, environmental decision, which is made at the level of nitrite, is really significant. Because when you fertilize your fields and your ammonia nitrogen first gets oxidized to nitrate and then denitrified, it's lost. You don't want that, right? You want to keep it on the field. So on the field, you would rather have ammonia. When you think about tinkering with natural ecosystems, sometimes in the context of global warming, people consider modifying ecosystems to do things differently. Well, if you could kind of stimulate ammonification rather than denitrification, you would stimulate primary productivity. whereas denitrification constrains primary productivity because you remove the nutrient from the system. So this is let's say, the choice that was one of the focus points in Bremen. And there was some very nice work on this topic. I guess for us, this paper was the most influential one by Tietje and co-authors, where they say, okay, well, when you look at it thermodynamically as denitrification, you see that denitrification has the highest amount of protons translocated over the membrane per electron. So per, let's say, electrons, or let's say per molecule of acetate, if you're oxidizing acetate or glucose, denitrification would give you the highest yield. Whereas ammonification gives a lower yield per electron, but because you have 6 electrons, you can donate 6 electrons to the nitrogen. In this case, the overall yield is higher. And so based on that, Tietje predicted that if the system is under electron donor limitation, you would get denitrification, and if you are under electron acceptor limitation, you would get ammonification. Okay, so thermodynamic inference, very similar to the redox tower, along the same line of thinking. There's been a lot of environmental studies, but they all give different results, and a lot of parameters were correlated, so it was not very helpful. Now, some people have approached or are approaching this problem by using pure cultures. So you could take a culture of a denitrifying organism and an ammonifying organism, put them together in a let's say laboratory mesocosm or chemostat or whatever, and then tweak the condition and see what happens. But I think that experiment is not good because you don't know very much about these organisms. And they're probably from very different habitats. So you never know when you see a change, like one is winning or the other, whether it's really related to this specific point. There may be some other factor. So the way we approached it was by using natural communities. I expected a different slide here. Okay, so what we did was, so Bremen is close to the North Sea, and the Wadden Sea, which is a big, let's say, estuary of much of the agricultural runoff of all of northwestern Europe. So there's a lot of nitrate going into this coastal ecosystem, and there's all these sandbanks. And the sandbanks are above the water during low tide and below the water during high tide. And it's sand, so it's a very high porosity, so the water really flows through these sandbanks, and they act as a kind of filter. So there's very high rates of both denitrification and ammonification in these ecosystems. Denitrification is dominant, but there is also ammonification. So that was the starting point. So we took those sediments, and we put them in our cement mixer to shake off the cells, and then inoculated chemostats with these cells. We did a lot of controls to show, we preserve a lot of the biodiversity this way, we preserve the activity, so that's the way we did it. Of course, you may argue that the chemostat is very different from what happens in the natural system, but that was really not the point. We were just interested in what is the condition that drives this choice. So Beate Kraft, who was the student who did this, she started with these incubations. And of course, we focused in the beginning on the carbon to nitrogen ratio, because if this ratio would be high, we would expect ammonification, according to Tietje. Now what you see on the graph here is the conversion of the nitrate, which is 100%, plus the organic nitrogen we supplied, because they also got amino acids. So 100% of the nitrogen going in, and what you see is how much goes out in the form of ammonium. So what you see in the graph is that the amount of ammonium that we generate is actually less than the organic ammonium we provided in the medium. So that means this experiment shows no evidence of nitrate reduction to ammonia. It only shows some ammonification of the amino acids. So then they have to increase the C to N ratio. So we have more organic nitrogen now, but again, she didn't see any ammonification. And then she increased the C2N ratio even further, far beyond what should be necessary, and again, we didn't find any ammonification. So this concept was at least not the complete truth, or the complete, let's say, picture. So she went further. Now, people said, well, maybe half a day of generation time is too short. You have to use a longer generation time. The in situ generation time was around 10 hours, we estimated, based on the in situ metagenome. So we went to 1.7 days, but that didn't make any difference. Then we started trying all those factors that have been identified in the literature as being important in natural ecosystems. Low pH didn't have any effect. No copper. Denitrifiers need copper, and ammonifying organisms do not need only iron, but copper also didn't make a difference. Natural seawater. In the other studies we used artificial seawater. You start to do trivial controls. Also didn't make any difference. Feast and famine. So providing the substrates not continuously at very low concentration, but with pulses, so that there was no competition for affinity or whatever. Didn't make any difference. Sulfide was in the literature as an important factor stimulating ammonification. Didn't make any difference. Fermentation products is also one of these things. Didn't make any difference. In all cases, we found denitrification. Yeah, so it was not so easy as hoped. And then Beate did one experiment that I did not think would give any result at all, so I strongly advised against it, but she did it, and lo and behold, that was the experiment that gave the ammonium. Yeah, and the difference between this experiment and all the other experiments in this graph is that she used nitrates instead of nitrites. So in all the experiments we used nitrites because based on thermodynamics, the effect predicted by teachers should be stronger with nitrites. Using nitrates, we got ammonification with quite a, not excessive C2N ratio. So that was quite surprising. So then we went further and Beate transferred the culture to a bigger chemostat so that we could study it in a bit more detail. And the first thing that happened was that the ammonium disappeared again. And then she realized that she had a lower generation time in her bigger chemostat than in her smaller chemostat. So when she increased the generation time again, so decreased the growth rate, we got the ammonium back. And this was a very interesting effect, we thought, so we played with it a bit more. And so then we decreased the generation time again, we went back to denitrification, and increased it, went back to ammonification. There was a kind of a back effect here, which was overcome by increasing the generation time even further. So there's a tipping point somewhere. In our experiment, it was around 1.7 days. So there was a kinetic effect here, which could not be predicted by thermodynamics. Now then we went into the molecular details and did DNA, RNA sequencing at different time points when there was denitrification or ammonification and proteomics. And you can see some of the intermediate results here. So you get your metagenome, you do the assembly, and then you have your assembled contigs, but then you want to know which contig belongs to which organism. And that step is called binning. And at that time, there was not a good publicly available binning program available, so we developed one, and you see here some output of that program, and you see all these different clouds in your metagenome of contigs. And what you see in the graph is the sequencing coverage, so how often was every base sequenced in the contig, so the depth of sequencing and the percent GC. And the program is a little bit more sophisticated than that, but you see already visual that you have all these clouds. All these clouds correspond to a particular population, a particular organism. And this way, by binning everything apart, we got like provisional whole genome sequences of all the dominant members of the culture at different time points. So these were the time points we sequenced, and here you see different denitrifying populations that were identified, ammonifying populations, and so one population that could only reduce nitrate to nitrite. So all the analysis of those metagenomes and some isotope labeling follow-up experiments by Beate showed that this is the way it goes. We provided glucose, amino acids, as the carbon source. Those were fermented to fermentation products, acetate, formate, hydrogen, by specialized populations. So these organisms did not take part in respiration. They were also not correlated to any particular condition. You could find them at any condition. Denitrifying or ammonifying, they didn't care. These fermentation products were then used to oxidize the CO2 by the respiring organisms. And of course, the final electron acceptor was nitrate, but Beate found a very nice cryptic sulfur cycle in between. So actually the sulfate reducers were better competitors for these compounds than the nitrate reducing organisms. So the nitrate reducing organisms used these compounds and sulfide as electron donors to reduce nitrate to nitrite, and then onwards to nitrogen or ammonium. Yeah, so that was a very different from the original redox tower I showed in the beginning, where denitrifiers are supposed to use, these compounds directly. And what are the important factors now? Well, the ammonification has higher fitness with nitrate, so not with nitrite, at high generation times, and I didn't show those results, but the experiments were also done. The carbon to nitrogen ratio is actually important. If you're under electron donor limitation, you will always get denitrification. Yeah? So these are the rules. If you want to tweak this competition to your advantage in agriculture or in souring control, these are the things you need to pay attention to. Of course, generation time is a tricky one, because how do you control that? The reason why, based on our experiments and analysis, the reason why it is this way is that the nitrite reductase of the ammonifying organisms has a lower affinity for nitrite. And when you look in the biochemistry literature, there is no trend in that. The measured affinities of the enzymes go in all directions. But when you think about it, it seems to be the most, let's say, parsimonious explanation, that when things go so when you use nitrite directly, so when the competition is for nitrite, the ammonifiers always lose because of this lower affinity. When you use nitrate, they use the same enzyme for nitrate reduction, the periplasmic nitrate reductase, and so they have equal affinity for that one. And then they produce the nitrite, but they produce it very close to their cells. So they have the advantage there, because if somebody else would like to use that, it first has to go by mass transport to another cell. So here, it's not bothered by this slightly lower affinity. And then when the carbon to nitrogen ratio is high, the thermodynamic advantage of the ammonifier pays off. So I think I have the feeling, it's for others to follow up, but I think this is a really nice concept of how to tweak. So now we know how to tweak the community to do something that you want. Okay. Oh, I actually included those slides. Can I talk until 4, or should we need some time for questions, right? So 5 more minutes. Is that okay? We can do. Okay. So this is a different. No, I mean now I'm going to talk for 5 more minutes, and then we can do the questions. So we'll finish at it's okay. So this is another study which has not yet been published, and it shows how these communities are actually assembled in a chemostat. So this chemostat was fed with oxygen twice daily. It is a denitrifying chemostat that receives oxygen twice per day, mimicking a tidal cycle of these tidal flags. Then you see how the populations developed and were selected during this incubation for these conditions. And you see that actually after about 50 days, there's still some dynamics, but the communities or the populations remain pretty constant. When you look at what happens during this tidal cycling, what you see here is the exposure of the culture to oxygen at the beginning of these 12 hours. So this is after about 100 days of these cycles. You see the oxygen concentration goes up, and then we measure the fate of the isotopically labeled nitrite, and we see that we get production of nitrogen right from the start. So that's called aerobic denitrification. It's not supposed to happen according to the redox tower, but many people have observed it. Because nitrate is less good than oxygen, right? So you don't expect the organisms when they would behave rationally to do that. Then when you look at the fatty acids, what you see is a peak of formate during the aerobic parts. Yeah, also rather counterintuitive because formate, you kind of reduced product. Why do you see a buildup of an electron donor in the most oxidized part of the cycle, during the most oxidized time? So that's quite interesting. Then we did the whole metagenomics again and analyzed how much different populations contribute to respiration. And this is also quantitative to the amount of substrate added. So you see oxygen was about a third of the total electron acceptor provided to the system. So we have oxygen reduction, we have sulfate reduction, nitrate, nitrite, denitrification, everything. And the four organisms are, the tasks, let's say the functions, are very much distributed over those organisms. So all populations in here compete for the oxygen. Also, the Vibrio, which is actually kind of otherwise fermentative in this complex. So it performs fermentation via pyruvate formate lyase. And even the disulfovibrio reduces the oxygen. These experiments were done by Jian Wei Chen, so one of my postdocs in Bremen, and he then incubated these cells with at different oxygen concentrations to go more into this phenomenon of aerobic denitrification. Now what you see is that in the chemostat the cells formed kind of small aggregates. And what he did was, and so what we thought was probably the reason they can denitrify is that they have oxygen limitation inside these aggregates. So there's actually very low oxygen concentrations in here. So he first filtered these suspensions to get suspended cells, and then you see there's no aerobic denitrification anymore, and you entirely lose it until you go beyond 50% air saturation. Then suddenly you get denitrification again with N2O as the end product. How can that be? Well, what I think is when you have oxygen limitation, you have a branched respiratory chain. So the electrons go to the CBB3 terminal oxidase, to the denitrification enzymes, and all is good, and the two processes work in parallel. When you go then add oxygen, denitrification is outcompeted for electrons. And now the oxygen reductase has plenty of power to absorb all these electrons, and you only get aerobic respiration, and denitrification stops. But then, when the oxygen concentration becomes very high, the terminal oxidase, which is CBB3 type, high affinity, gets inhibited by oxygen. And now, the denitrifying enzymes suddenly become important again, except for the final one, which is, inhibited by oxygen or deactivated. The N2O reductase is the only enzyme of the denitrification pathway which is intrinsically sensitive to oxygen. So you get N2O as the product. Yeah, so that's another example of how, let's say, I'm going to skip this part now, of how the game, you have to understand it before you can tweak your community. So back to the microalgae. So what I'm doing in Calgary is this is kind of the conceptual approach. So we design a theoretical full-scale process that reduces the cost to the bare minimum. So I don't care about what's in the textbooks. This is what I want. These conditions are ideal. That's where we can reduce the cost. Then we go to a natural ecosystem that's similar to this process and find our community and apply natural selection of the community to make the whole thing work. And so that we go with the force. Yeah. Okay. So then acknowledgement. So I summarized a lot of all the work from the TU Delft and the Radboud University in Nijmegen, the students, Boran, I showed some of Ashna and Katarina. Yes. Okay. Then the students at the Max Planck Institute, students and postdocs, long-time collaborators. Oh yeah, here are my mentors. Guys couldn't my job. Okay. Thank you very much. Yeah.
You mentioned that the solvent reducers outcompeted the nitrate reducers for hydrogen forming, which I found surprising. Can you have any idea why that is?
No, I don't have any idea why that is. But it's kind of consistent with the field observations, right? I didn't go into that slide, but so when you look at there's a very nice study by different people, one of them Canfield, about sulfate reduction in the oxygen minimum zones, where you also of the ocean, the dead zones. When you look at the gene counts of the genes for aerobic respiration and sulfate reduction, they're about 1 to 10, both in activity, transcriptome activity, and abundance. In the two habitats compared. Those were exactly the ratios we found in our bioreactor. So it seems to be a general phenomenon. But why, if you would ask me why fermenters outcompeted denitrifiers for glucose, I have a theory about that. But for sulfate reduction, because I don't know why they're so good. Yeah.
Mark, I had a question, Stephen, you said a few minutes ago about the nitrite reductases saying they have low affinity for nitrite.
Yeah.
When they're producing ammonia, but a lot of NRSA, NERAS, et cetera, have KMs in the low micromolar, 5 to 50 micromolar.
Yeah. So what I meant to say was that the nerve system has a lower affinity than the NERS or NERK system. Yeah, that was what we hypothesized in this paper, especially when growing with mixed substrates. So this difference, so we couldn't measure affinities with our communities, but we could look at the maximum conversion rate, and the maximum conversion rate for denitrification was especially higher when the cultures were provided with sulfide and acetate as the electron donors. So I think that the bottleneck is probably in the electron transport towards the nerve A, maybe more so than what the enzyme could do if provided with unlimited electrons. And I guess that was what I was asking you during lunch. Do you know how the electrons get transferred into this enzyme? Because you need 3 pairs. So there may be some logistic bottlenecks in there. And I think the difference is very slight, because you see how long it takes for these communities to go and move in one direction or the other. It's not like a huge difference. It's probably a pretty small difference in affinity.
Yeah, I mean, it has to be a very small difference because NRSA is very high affinity for...
Yeah, but NERAS too. So when you look at the literature, we looked at all the studies on that presented affinity values and found them to be very variable. That was one thing. Some people mentioned pretty poor affinities, but others mentioned really low affinity constants. So we couldn't say based on that whether one enzyme had better affinity than the other, but our results pointed in the direction that the nerve has the poorer affinity, even though it may still be very high. But, yeah.
I have a question about. So, as far as controlling, if nitrite controls the end product, the amount of nitrite that's being added to these systems, what controls nitrite fluxes into natural ecosystems?
Yeah, that's right.
Thought about what?
Yeah. That's right. So one of the factors. So what controls nitrite in natural ecosystem? Well, it's the balance between the nitrifiers, let's say the first part of nitrification, where the organisms oxidize the ammonia to nitrite, and the second part, where the nitrite is oxidized to nitrate. If that ratio changes, nitrite or nitrate may become the more dominant electron acceptor for denitrification. Now, what kind of factors could change that? Well, for example, sulfide. Sulfide is one of the factors I think that can be pretty inhibitory for the first step. So if you inhibit the first step with sulfide, you may get more nitrate as the end product, because now the first step is the controlling factor, which could that way stimulate ammonification. Yeah.
Are there natural systems where this can be observed?
No, I think when you look at the natural ecosystems, well,
sulfide fluxes, I guess.
Yeah, so that's right. So it's pretty difficult to do this with the natural ecosystem. Yeah. It's just pretty difficult to control. So that's also why you have all these factors and they do correlation analysis. Well, you see also in our study that this cryptic sulfur cycling. Go back to the. Yeah, this cryptic sulfur cycling is more intense with ammonification. So the system is a bit more reduced. So actually you can measure a little bit of sulfide. With denitrification you don't measure it, but still it's 25% of the turnover. So that's why it's cryptic. I guess the difference between a flux and a concentration is very important. And a flux is really difficult to measure in anything, but in the oceans they can measure that with very extended gradients. Like oxygen gradients that persist for hundreds of meters, or in sediments for meters, then you can calculate a flux. But in a soil
Kind of just to make sure I'm understanding maybe what I think is a key concept is related to what you just said, the flux. So does that typically explain, like, unless there are other sort of more subtle, funky things going on, why like not the most thermodynamically favorable pathway takes place, but because the flux, like fermenters, if they just go to formate instead of something more, oxidized, then they can divide more, like basically like over time, like energy over time is higher.
So I think if I guess I'm not sure if I'm going to answer it right the first time, but follow up. So essentially kinetics is more important than we like to believe. We don't like to believe kinetics is important because it's much more difficult to determine experimentally. Thermodynamics you can calculate, right? So kinetics is a really ugly thing in your model. But it looks like kinetics is important. So the barrier to do a chemical reaction depends on the activation energy. And these those reactions that have high activation energy are at a disadvantage, or have some step, like some reactions are complex, right? Like nerve has different kind of activation barriers it has to follow. So the activation energy kind of is the one that screws it up. And then fermentation, I think in fermentation things are kind of simple because these activation energies are not so high. So I think in fermentation you can do more with thermodynamics. Also you're closer to equilibrium compared, like these respiratory processes are so dissipative. It's very much non-equilibrium thermodynamics. So not easy to say anything. But fermentation is close, near much closer to equilibrium. So it's more what Prigogine would call the near equilibrium regime of non-equilibrium thermodynamics, where things are in principle still linearly correlated to Gibbs free energy changes. Yeah.
So I may have missed this. I'm sure it's obvious, but you said sulfide is inhibitory to
nitrification.
Nitrification. Yeah. Why is that?
Why is that? You shouldn't ask me. Is there a nitrification specialist in the audience? I guess it's the ammonia monooxygenase, but I'm not sure. Or, I'm not sure. Sulfide is also very inhibitory for anaerobic ammonium oxidation. And I also don't know why.
Any more questions? There's some time if you have a question to ask Mark after the talk. Let's thank Mark one more time.